Yeah, hopefully we'll be able to get a more cohesive uh, grouping of information for class. All right, now I'm gonna go ahead and jump to um, the syllabus. And we're going to talk a little bit about it, or the disaster relief resources. Let's do that. Let me go ahead and screen share. Everyone should be able to see, and we're going to jump straight into that. So if you go here on Google, you can look up disaster resources at App State. So these are university provided resources, and also have some information about community provided resources. So you can see here, we have some information about FEMA. There are 3 ways to apply. Online, you can download the FEMA app or call. So go ahead and you're going to upload documents to images and photograph them. So we have the Red Cross emergency shelter information if something is going on, non-emergency phone numbers if something is going on. There is counseling resources. And if you are experiencing an urgent mental health concern, you have the number here. Please call them. They have free, informal, and confidential consultations with a counselor, no appointment needed Monday to Friday, and drop-in support and connection following Hurricane Helene. So that's in Roan Mountain, the Plymouth Student Union. And they have additional mental health resources here that you can click on. So New River Light and Power is the company that controls most of the electricity outsourcing for Western North Carolina. They have a website here, Facebook, Instagram, and you can find their outage map and information about disconnecting service. You can get in contact with them that way. Their customer service hours are here for how to speak to representatives. Here's some places on campus that have internet access: Belk Library, the parking deck, Central Dining Hall, College Street parking deck, Plymouth Student Union, and Sanford Mall. They have a Mountaineer food hub and free store available for faculty, staff, and students. And you can go ahead and check that out if you need to. That link is here. We also have some academic resources. Here's logistical support and resources for the resumption of classes, resources for the return of teaching and learning, Office of Student Success, and you can get in contact with them if you're struggling about anything. Community resource information from the town of Boone, the area of Chamber and Commerce, UNC Health Appalachian, Catawba County, and then the Chamber of Catawba County. There are also some volunteer opportunities for those of you who were not impacted, if that's something that you would like to do. We have the Appalachian Medical Reserve Corps. There are also donation opportunities to the Disaster Relief Recovery Fund, the Boone Area Chamber of Commerce Foundations, and the North Carolina Disaster Relief Fund. So you can find that information there. And again, if you click on each one of these, it'll kind of just take you there. Some other things that I did want to bring to your attention that are not mentioned on here is I think it's up until maybe tomorrow or the day after. If you have any kind of food insecurity as a result of Hurricane Helene, you can apply for disaster food stamps. The FEMA processing, from what I have heard, can take a while. I've been informed, to let students know they take very, very clear photos when you upload them into the portal, because they are using fax machines to get information back and forth across. So just a couple of things about that. And I also would highly and very strongly recommend, if you have not gotten in contact with the Dean of Students, if something is bothering you, please do so now. They have a disaster relief recovery hub. And for some of the students who are attending that who have been heavily impacted, especially those who live off campus in apartments, because I think it was over 120 apartment complexes that were condemned. So you can get in contact with them and they can help you with any kind of financial issues you were struggling with. And again, my student support hours are open. I can see online whenever you guys join the hours. So if you join and the meeting is not open, I can see it on my email account and it sends me a notification so I can start the meeting. Other things, you can also email me, especially if it's anything directly related to academic performance and thoughts about struggling as a result of that. And we will work with you. And we're going to talk a little bit about how we'll work with you, the things that we can and cannot do, as well as academic changes that have happened to the syllabus in order to promote student success as a result of Hurricane Helene. So let's go ahead and click here. If you go to the dashboard, you click Bio 1801 Labs Fall, and then you scroll here. The updated Bio 1801 syllabus is there. And I'm going to go ahead and upload this or look at this in a new window. So, so here, there are some adjustments that have been made due to the school closure. And again, we want you to know that our biggest focus on the safety, health, and concern for, you know, we want you concerned for yourselves. And we know that it's going to be hard to return, and that is why we are here to help you and make accommodations and work with you if something is going on. Um, that being said, you do need to let us know so we can work with you, because certain accommodations are only not being offered. So we're going to scroll here and kind of look at some of the changes, the biggest ones, like workplaces that have been added for students. If you're having, and the other thing too, I wanted to mention about internet access issues. Some local libraries, depending on the county, let you check out internet access. Small portable modems for you to take home, and it's worth checking with your local library if you're having issues with that. So again, professionalism points is now 90 points. You are being offered professionalism points at this point in time, no matter what. That could change later on. We're not sure. Pre-lab exercises are weighted now at 215 points. There have been some assignment drops with that. Digital lab notebooks with your lowest grade dropped is now 8 DLNs at 245 points. Your post-lab quizzes with the lowest grade dropped is now 140 lab points. Your module projects are now 2. So it will be the module project you've already completed, and then the genetic diseases module project at 120 points. And that is a total for 840 points instead of 1,000. So things are being weighted more heavily. That being said, you can see the end of semester surveys are still there. Make sure at the end of the semester, I want every single student to do those. This has been a rough semester. Please utilize your resources. Please go ahead and make sure you get as many points as you can. Those are free points. Take them. And again, the grading scale is consistent. Here, again, some details about that. Again, now our attendance policy. So we are so we currently do not know whenever Rankin Science will be opened back up. It could be the end of the month. It could be the end of next month. We do not know. It may not open up at all this semester. That being said, for the time being, we are doing virtual work. So we are offering extensions that have to be turned in within 7 days of the initial submission date, but we need to be aware of it. And it is only being offered on a case-by-case basis, which again is why I am asking you guys to reach out to me and let me know if you're struggling with anything. That way, if you have questions about opening assignments and extending assignment deadlines, I can refer you over to my supervisor with information about that. So, again, assignment deadlines and late work policy. Yeah, that being said, the deadlines are set for the week of November 15th to 29th are final. So everything after 15th to 29th, we cannot offer extensions for because it is too close to the end of the semester. Last day to drop is August 23rd, and last day to drop music career drop has been extended to November 8th. So you will be getting your grades in at some point before November 8th. That way, if you decide that you do not wish to stay in the class, you are able to utilize that to your best knowledge. And a little bit more information down here, you can kind of see a tentative schedule, and this is a tentative schedule of what the end of the semester is going to look like. Here we have that Helene closure, the fall break assignment submission, and you've got information here. And then, a little bit more information as the semester comes to a close here. But everything from this submission deadline forward cannot get an extension for the week November 18th down. So please be aware of that. There cannot be extensions made at that point in time. I do want to take a moment to go ahead and review the PowerPoint slides with you all, so you can kind of have an idea of that. And again, if the internet access cuts out, please let me know. And again, it has been a little bit shoddy in and out, but we're going to review some of this information. If you have not watched the procedure video, I'll pause for 8 minutes whenever we get to the methods and go ahead and have you watch it. That procedure video can be found here on As You Learn for this week. So, yeah. Should be week 10. There you go. You can kind of see that procedure video here. And this procedure video, as you are watching it, when we get to that point, I would like for you to go ahead, open the Post-Helene disaster spreadsheet that was provided for you on As You Learn, and include the information that was recorded by the Bio 1801 team here in these points, because you're going to pretend kind of like you're working with them, almost as if you're part of their group. So, the respiration, where glucose and oxygen produce carbon dioxide, water, and ATP as energy. During this process, there are a couple macromolecules of interest, enzymes that catabolize, and remember cat, breaking down, molecule reactions in the biochemical pathways of fermentation. ATP, this is something that the cell uses to create or maintain having energy stores. It is a nucleic acid, okay, with an adenine base and a pentose sugar, and then 3 triphosphates. And you can kind of see that here. You have ribose, which is that pentose sugar, adenine, and those phosphate groups. Glucose monomers are the building blocks of macromolecule polysaccharides, such as starch and glycogen, that are important to this process, and you can kind of see those here. Now, in order to understand fermentation, you first have to understand aerobic respiration. So it includes 4 main systems. You can see glycolysis here, the Krebs cycle, and the electron transport chain, or 3 main systems. In glycolysis, glucose is broken down into pyruvate, producing ATP. Brain of the mitochondrion through the cytosol into the Krebs cycle. Okay? When glycolysis occurs, electrons are carried via NADH into the electron transport chain. The Krebs cycle is a cycle that is very long and includes catabolism of an enzymatic reaction in order to create ATP. And it also releases electrons via NADH and FADH2 into that electron transport chain. When you think of the electron transport chain, I almost want you to think of it as steps, like you're stepping down a step of steps, okay? We are looking at molecules that are carrying electrons in a downward motion, okay? And as they are carrying these electrons in a downward motion, they are moving towards oxygen, okay? Oxygen is the last step at the end of those steps, and it is the electron recipient, okay? So anaerobic respiration is a type of respiration that can occur in the absence of oxygen. It is very similar to aerobic respiration, but it has a significant difference at the end. Oxygen is not the electron transport chain recipient, okay? So we're going to talk a little bit about that, because anaerobic respiration can be synonymous with the term fermentation. So fermentation only includes glycolysis, okay, the process of respiration paired with another step to regenerate ATP. And again, glycolysis is where glucose becomes pyruvate, okay? Therefore, it only uses substrate level phosphorylation of glycolysis to produce a very limited number of ATP. There are two main versions of glycolysis that can occur: alcoholic fermentation and lactic acid fermentation. It must repeat for more ATP, but repeating glycolysis is quick and easy. Fermentation occurs when ATP is needed quickly. So, in mammals, when the process of fermentation occurs, it typically occurs again when there's an absence of oxygen. A way to remember this is, how many of you guys run track? Okay, or do sports? You can go ahead and speak out loud for me, okay? Do any of you guys do sports or have any history with sports at all? You could just say yes if you do. I know a couple of you do. Now, when you are doing sports, if you work out or if you're running, okay, you get that kind of burn that happens in your legs. It's a little bit painful, right? That's because we're producing alcohol and lactic acid as a result of fermentation. It's a way to get a certain number of ATP really quick when you don't have a lot of oxygen coming into cells. So remember that. And alcoholic fermentation is a process where glucose becomes pyruvate, right? We're regenerating NADH and forming ATP during that process. Okay, regenerated ATP is then further NAD is regenerated into, yeah, we have NAD plus that becomes NADH again whenever pyruvate is turned into ethyl alcohol and carbon dioxide by the enzyme alcohol dehydrogenase in the process of alcoholic fermentation. Pyruvate can also be broken down into lactate by lactate dehydrogenase, which also helps regenerate NADH during lactic acid fermentation. So these are two processes that yeast can do. Okay. Yeast as a model system includes the use of the organism Saccharomyces cerevisiae. Let's say that again. Saccharomyces cerevisiae. Can be hard to say. Saccharomyces cerevisiae. It's a single-cell fungi used to make bread, beer, and wine, and is an exceptionally well-studied model organism. So if you know anyone who has gone to undergrad, I don't know if you know this or not, but brewers or people who make beer actually have to go to school a very long time to do it. Um, especially these people who work with these really large companies, and they have to have a very good understanding of fermentation, especially what alcoholic fermentation. So, again, this is something that is very common. It's all over fruits and vegetables and grapes. The recipe for beer is almost 4,000 years old, so we know this process works, and it has a fully sequenced genome. So, and again, yeast converts sugar during the process of fermentation to carbon dioxide and alcohol during alcoholic fermentation. Okay, and it can actually ferment even in the presence of oxygen. Okay, that's not as something as common in other organisms, right? And this is known as the Crabtree effect. In the presence of high oxygen and sugar, yeast will use both fermentation and respiration. We're going to talk a little bit about that. Baker's yeast, for that reason, is an excellent model for fermentation. Remember that when you're working through your DLN, okay? Alcoholic fermentation in yeast. Glucose is imported into the cell by hexose sugar or hexose transporter via monosaccharide transmembrane transporter. Okay, and it goes through a conversion into fructose. Fructose becomes glyceraldehyde, 3P, and then it goes through a 10-step glycolysis to produce, or it finalizes at 10 steps of glycolysis to produce 2 units of pyruvate. So for every unit of glucose, 2 units of pyruvate. Okay, which means 4 carbon dioxides and 4 units of ethanol. Remember that. Okay. In the final stages of fermentation, this pyruvate molecule is converted into acetaldehyde and carbon dioxide is released. Okay, and because we're going through it twice, it's 4 units of carbon dioxide, and which is then converted into ethanol by the enzyme alcohol dehydrogenase. Remember, we talked about that. Both ethanol and carbon dioxide are highly toxic to the cell in high concentrations and thus are exported out of the cell. And remember, that's why we feel that little bit of a burn. So we're going to talk about alcoholic fermentation in the processes of food metabolism, especially within Saccharomyces cerevisiae. Food sources produce ethanol and carbon dioxide with the extrusion of ATP. When you have better food sources, you have more products, carbon dioxide and ethanol. So when alcoholic fermentation occurs and you have food versus no food, what does that look like whenever Saccharomyces cerevisiae is exposed to these environments? So when no food is present, yeast and water, it does have intracellular food stores, okay? And a limited background level of fermentation will occur. Small amounts of carbon dioxide and ethanol will be produced. Whenever you have yeast in the presence of food, in this case, glucose, and that is a known food source, significantly more fermentation occurs when the food is present in the environment, and large amounts of carbon dioxide and ethanol are produced. Okay? So, if we're looking at the production of carbon dioxide as a result of fermentation in the presence and absence of food, okay? Question. Does structure compare yeast and glucose, and it's a known food source? We're going to talk a little bit about hypothesis states that structure does not determine function. Our alternative hypothesis states that structure does determine function. It's important to remember when we're talking about structure, we're talking about how something is arranged to allow it to perform a specific job, task, or function. Okay? So it's something that has been relatively proven within the scientific world. And we can't necessarily say anything is proven per se within the scientific world. It's just highly probable, especially if multiple research papers have been done and all of them point to the same answer. So when you're thinking about this in class and you're thinking about the hypothesis, I want you to remember this is something that has been highly established within the scientific world, and we're going to talk about that and why. So here are a couple of examples of how it's highly established. There are two basic shapes for proteins, fibrous and globular or round. Fibrous proteins, like collagen, are shaped like a rope and they give a bit of strength to our skin to prevent it from tearing. Globular proteins, such as hemoglobin, are used to transport oxygen in blood. And again, that shape is directly related to what they do. That proves structure and function. And then we have cellular anatomy and how cellular anatomy determines its function within an organism. Neuron cells are really long. Okay, you can see that here. And they send messages throughout the body. Fibrous shapes of skeletal muscles allow for movement by contracting. And again, that proves structure and function have a very established relationship within the scientific community. And again, we can talk about anatomy and physiology even at the animalia level. So, anatomy is the structure of the body. Physiology is how the body functions. The anatomy of bird bills determine their food sources. Look at this. We have a bird that has a basic beak. It's a generalist. One with a short, sharp beak for insect catching. A probing beak in order to probe for food. Surface skimming. You can see that flattened in. Mud probing. Filter feeding. Pursuit fishing, aerial fishing, fruit eating, nectar feeding, chiseling, dip netting, scavenging, raptorial, that's for hunting. And all of these shapes are related exactly to what they do. It is something that is observable within the scientific world, okay? So here we have two molecules. I know seeing these molecules on a screen, especially with all these letters, it's like, oh my gosh. What you need to know about these molecules is glucose and galactose are stereoisomers, meaning that they have at least one group that is an opposite orientation of each other. Here, that group is an oxygen and hydrogen, okay? In glucose, that oxygen and hydrogen is located below carbon, okay? In galactose, it is oriented above. So I know you guys may not have taken something like organic chemistry at this point in your academic careers, but I do want to show you something in order to emphasize to you the importance of stereoisomer orientation, chiral molecule orientation, and function. These molecules will not process in a chemical reaction if they are not shaped a specific way. Okay? And we know that glucose can ferment. But do we know that about galactose? It's very unlikely. So let's look at the molecular orientation of two chiral molecules. Just so you can kind of see something really quick. Okay. It's not happening unless you flip it over. Okay. If you try to put it on as it came out, the way it would fit your right hand, it would not go on. You would have to turn it. And these chemical reactions don't operate that way. Okay. So let's go back to the lecture material. All right. So for our experimental design, we have yeast and water, yeast and glucose, and yeast and galactose. We know that yeast and water, even though it doesn't have a food source, is going to produce a baseline amount of carbon dioxide as a product from this reaction. Okay? Therefore, its baseline amount of carbon dioxide produced, yeast and glucose has a known reason. Okay? And not only is for research papers, side output, we're talking about isomers and we're talking about the ability to work those. If structure, yeast and glucose and yeast and galactose, we both lactose and yeast and glucose have differences in structure that impact function, then it's not going to be able to produce carbon dioxide because it's not shaped like something we already know about. Therefore, we know about yeast, it's going to function as a control group. The lab we did before break, we had a positive control that was also our control group. And remember, there is a difference between a positive control and a control group. Yeast and galactose, okay, is going to be our experimental group because we don't know the outcome of the experiment whenever we are testing, right? We have anticipated results, but we're just testing something that's already known. So if structure does not determine function, yeast and glucose and yeast and galactose in this experiment would produce the same amount of carbon dioxide because they would both function the same in this experiment. But remember, you can't really put a left hand glove on your right hand unless you flip it. And that's really what I'm trying to drive home here. These chemical reactions cannot occur if these molecules are not oriented in the perfect way. So we're anticipating that structure does determine function, right? And we are anticipating that galactose is not going to be able to produce carbon dioxide. So, and again, we're tracking this through the production of that. And now that you guys are here at this point with the experimental setup, just say yes so I'll know that you're paying attention real quick. Yeah. So you guys are following along though, I hope. Go ahead and watch the because this is where I am right now with this. I think the screen sharing broke for a moment because the connectivity went down. Go ahead and go on as you learn and click here, okay? And scroll here and watch the lab procedure video. I'm going to sit here for a second. I'm going to put myself on mute, set a timer for 8 minutes. Go ahead and pull it up. That way you can watch that video, and then we'll move on from talking about the experimental setup, okay? Okay. So now we're going to come back to what we were doing, and I'm going to screen share again, and we're going to talk through the rest of these PowerPoint slides as a group, okay? So coming back to where we were, again, you're expecting that buildup of carbon dioxide. And again, here you have independent variables. Your independent variable is going to be your yeast media and whatever sugar or lack of sugar is being added in. Dependent variables are going to be the production of carbon dioxide and ethanol. Controlled and confounding variables. Your biggest controlled variable that we want to talk about here is temperature, right? It's being held at 37 degrees Celsius, okay? And confounding variables would include for example, the fact that we're not washing it with soap because it's toxic to Saccharomyces cerevisiae, there may be leftover yeast in the tube. And we have negative and positive control groups, and we talked about those. So our negative control group would be yeast and water, and our positive control group would be what? Can you guys tell me? What would our positive control group be?
Is it yeast and glucose?
Yeah, it is. It's yeast and glucose. So, and with our experimental setup, if we're talking about the experimental system, okay, and we're doing a deductive logic statement, and remember with the deductive logic statements, we're talking about comparisons of mean. We would expect the mean quantity of carbon dioxide produced by yeast and glucose to be statistically greater than the mean amount of carbon dioxide produced by yeast and what? Any ideas? It's our negative control. Yeast and water, right? So, right. If yeast and water and yeast and glucose are negative control and positive control, what would our positive control in our experimental group be? Or our control in our experimental group? Well, we know the outcome from yeast and glucose, right? And we need a baseline to understand the outcome with yeast and galactose in order to test whether or not the alternative hypothesis is valid. So it would be yeast and what? What would be our control group? Any ideas on the control group? It would be yeast and glucose. Because our positive control here is also our control group. And our experimental group would be what? Yeast and galactose, because it's what we're testing. Okay? And for our experimental setup, right, if we have our prediction, we can perform our deductive logic statement for the null hypothesis. For the null hypothesis, we're stating structure does not determine function. Okay? That would mean that we are testing to see if the mean amount of carbon dioxide between yeast and glucose and yeast and galactose is the same. So if the null hypothesis is valid, okay, then the mean amount of glucose and yeast and carbon dioxide produced by glucose and yeast is going to be statistically the same as the mean amount of carbon dioxide produced by what? Yeast and galactose. Okay, and for the alternative statement or alternative hypothesis, we would expect that structure impacts function. We know that yeast and glucose produces carbon dioxide. So if we're talking about structure impacting function, if structure impacts function, then the mean amount of glucose or carbon dioxide produced by yeast and what? Glucose will be statistically greater than the mean amount of carbon dioxide produced by what? Yeast and galactose. Okay, so this is what we were doing before. We were just comparing confidence intervals. Today, we're also going to be comparing p-values. So p-value confidence intervals, are based off degrees of freedom. There's a confidence interval for each bar, and we were looking at overlap between them to see what treatments we were interested in. It's based on the variance of the data, specifically the standard deviation and standard of error. And basically, our answer was just yes or no, whether it overlapped or it didn't. But we can observationally see how much overlap there was. For p-values, it's based on degrees of freedom, right? Based on the number of replicates impacts the value. More values increase our confidence. There's only one p-value per comparison. You can see that's why we have connectivity here with the p-values on top. One for the experimental system, where we compare our positive and negative controls, and one for testing our hypothesis, where we compare our control and experimental groups. Okay? And we're going to be looking at statistical significance, okay? If the p-value is greater than 0.05, the two samples are basically the same. They're more likely to occur, and thus 95% chance of occurring, meaning that results are not statistically significant. The differences are not significant. Remember this when you're writing your lab notebook, because you want to put that in there. If the p-values are 0.05 or less, the two samples are different. They're just less likely to occur and thus only have a 5% chance. And that is establishing statistical significance, okay? So for our data that we're going to be using for class, because it's already been provided, there are no outliers, okay? But if there were, you might want to include them in the data set and discuss that within your DLN, right? So consider the analyzed data. Which groups are statistically different from one another, which are not? Okay? Right? What do you expect the p-value to be? Is that what you observed? Okay? And we're going to talk a little bit about generating figures now. Now, if you have not pulled up the spreadsheet that I sent you, please do so now. Okay? I sent this out to everyone, so everyone should be pulling that up right now. So when you watched that video, you were recording the results here, okay? Then you took those results and you put them in here. For the generation of the figures, I went ahead and put 0.1, 3.8, and 0.1 here. Okay? Yours may not look the same, but this is a basic idea of what they should look like, right? I took this data, highlighted it. Well, I'm hoping I'm still screen sharing. Let me check real quick. I am. Okay. I highlighted this data. I went to insert, okay, chart, inserted a chart, unchecked this box, and now I have these figures, okay? And when I clicked here on these bars, and I just did this so I had a bar for comparison, I went down and I clicked here and changed this from 0.1 to 0.0, just so we had a bar, right? And I did that here as well. So, and after I had done that, I kind of just went and made it pretty, right? Like we've done this before. We add in the title, we put our data labels, we change it to colors cohesive, we put our mean values on top, okay? We put our independent variable here, our dependent variable here, with our Y label values and make it pretty. We have our grid lines, our major grid lines across, our minor grid lines on the outside, and it kind of just looks like that. In order to get this figure to look like this figure, this is what I did. And I did this in Google Sheets so that it was easier for you guys basically to kind of see what you need to do. I clicked this bar here. I went to download chart as a PNG file. Okay. And you can kind of see on the video that it's there in the corner. Then I went to insert Oh, give it a minute. Oh, my computer is being a butt. Then I went to insert and I went to drawing. Okay. In this drawing area, I clicked the image icon. Waited for my computer not to be frozen anymore. And then went to browse. Okay. And then I was able to open up the image I had just downloaded. And again, you'd want to do this with your pretty image, right? You can insert lines here in order to kind of show relationships, okay? And in that same right, you can take these lines and color coordinate them. Then you can take these lines and create basically a legend, right? And just kind of insert it in your finalized figure. And you'd want to put your experimental group, or your experimental p-value. You can just put experimental, and you can put what? Positive, negative, or yeah, your control and your, oh wait, no, experimental system, sorry. And you'd want to put like your positive and negative control, so it kind of looks like that. And you can do the same for when you're comparing your to get and wait, yeah, and you put your p-value in as well. So, and that's just a way for you guys to kind of make a figure in a new different way. And you can make it pretty and orient stuff to where it looks nice and kind of get your data in there. So here you can kind of see me backing out of that. And by the end of it, you should be able to have a figure that looks kind of like this, where you have your experimental system comparison with a p-value of less than 0.05, where you're looking at yeast and water and then yeast and glucose, and your experimental results significance, where you're looking at yeast and glucose and yeast and galactose. Again, negative control, positive control, experimental. Positive control is also control group. And in this spreadsheet, there are also instructions at the bottom on how to complete this to kind of help you. Now that that's kind of where we are, I do want to ask you guys, do you want me to just go through the whole DLN with you really quickly and discuss that? Or just parts of it? It may be beneficial to talk about the whole thing. What do you think? You go ahead and speak. You can just unmute yourself. Yeah. Yeah. Annika and Paloma say yes. Presley says yes. Okay. So, yeah, we can just talk about it. We'll blast through it really fast. Hopefully, this recording is a good one. The last two labs, it was kind of hard to get things sorted. So, I'm hoping that this one's the best and this is the one that gets uploaded to YouTube. So, we'll see. Okay. Now, let's go back here on the As You Learn course page. Okay. And now you guys can kind of just all open up and follow along with what I'm saying. I'm going to be going really fast. And kind of go to the DLN submission box. All right. Give it a moment to load. And now we're going to go to continue to the last preview for me, because I kind of showed this to everyone. And we're going to start at question number 1. And I'm just going to talk to you guys really quickly about this and check again that this is recording. Oh, no. Nope, it's still recording. Okay, good. All right, now, okay, consider the process of carbohydrate metabolism, respiration, and yeast. Yeast typically default to fermentation, specifically alcoholic fermentation, as their preferred mode of respiration. Alcoholic fermentation is glycolysis, which is an additional step to regenerate NAD+. The inputs are glucose and ADP, and the outputs are ATP, ethanol, and CO2. Describe the biochemical pathway of alcoholic fermentation. Split between the steps involved in glycolysis and the steps needed to regenerate NAD+. Be sure to include input, processes, chemical conversions, and outputs. What it's asking you to do is go here, open the slides up, explain this. Okay? Now, consider a biochemical pathway of alcoholic fermentation of yeast and connect that to what we will do in the lab. We will need to provide yeast during the experiment inputs. What do we expect to occur, right? We expect alcoholic fermentation to occur in a quantity where there is more ATP generated and carbon dioxide generated as a result of fermentation occurring in the presence of food. What outputs will we be looking for and measuring? CO2. Okay. Define yeast and describe how and why they serve as a good model system for studying alcoholic fermentation. You can kind of go here and input this information. Okay. Based on your background readings and materials we provided you, practice your research skills by finding a relevant scientific paper. I mean, you can find something even from Budweiser or like Michelob Ultra or something. They have a lot of research papers about their beers, and they use Saccharomyces cerevisiae or brewers used to generate that beer. And you can talk about it. Some of them have their own genetic lines of these yeast that they use, and you can talk about it. A lot of these companies do that, and that'd be a great way to find a paper. What is the question we're trying to answer in this experiment? It's been provided to you, right? Okay. Now we're talking about our null and alternative hypothesis. Okay. Alternative is that structure determines function. Null is structure does not determine function, right? Now, independent variable, the yeast and the sugar, right, that we've chosen to input, and why? Because it's going to have differing quantities of carbon dioxide produced depending on whether structure determines function or not. Because we're looking at yeast and galactose, which is a stereoisomer of yeast and glucose, and they can impact function in a different way. Dependent variable, the production of carbon dioxide, because it is a result of alcoholic fermentation of yeast and glucose and yeast and galactose. Okay? What variables will we control in the experiment? There are multiple, but the biggest one is what? You can go here, you can see it. Temperature and time. Okay? What variables may be out of our control and possibly act as confounding variables? Again, we kind of cleaned this using what? Without soap. We cleaned it without soap, right? Not to kill the yeast, but that means there's remaining yeast left inside. So that could be a confounding variable. Okay? You can talk about more. Phenomenon of interest, right? CO2 produced. Consider what the phenomenon of interest is of this experiment and identify it below. And when we say consider, explain why that's a phenomenon of interest. We're measuring it as a result of what? Think about that. Which group is a negative control and why? Right? It's going to be the yeast media, including water, because it's going to create low level or baseline results to compare to the positive control of the experiment, wherein Saccharomyces cerevisiae produces only that baseline result of carbon dioxide as an effect of having intracellular stores of yeast or sugars. Our positive control and why? Okay, yeast and glucose. It's been established as having a known effect, and this particular stereoisomer of this sugar that we are looking at has been tested and established to affect fermentation within what? Saccharomyces cerevisiae, okay? And it produces a higher level of carbon dioxide input in alcoholic fermentation. What is your prediction for the good working order of the experiment? Okay, and remember, prediction is a comparison of means. You need to give more detail, okay, because I'll only offer partial credit if you don't explain in the means, identify the groups, and give me those values. But we would expect what? Okay, a higher quantity, okay, of what? Carbon dioxide. A higher mean quantity of carbon dioxide produced as an effect of fermentation in Saccharomyces cerevisiae with glucose as compared to Saccharomyces cerevisiae and what? Water. Okay? And then that if then statement. If there is good working order for the experiment, then occasionally carbon of alcoholic fermentation occurs. Okay, what might actually explain this phenomenon within the negative control? Again, Saccharomyces cerevisiae has that intracellular yeast store, and you're going to talk about that. Okay, what negative and positive control groups results would lead you to believe that the experimental system is not working as anticipated? Well, if the negative control group produces a greater quantity of carbon dioxide as compared to the what positive control in this experiment, and identify what those groups are, would you continue on and test the experimental sample? No. Why or why not? Why? Because it's been contaminated, and you cannot interpret the experimental results whenever the experimental system is not operating correctly. And again, here we're talking about passive voice and past tense. There has been a document uploaded to As You Learn to help you with passive voice and past tense. Try not to use first person pronouns in these papers. Past tense is something occurred, not something is occurring when you're writing, right? And passive voice. So, again, passive writing. You can work on that. And you're going to record the methods you use to test the good working order of the experimental system and go back and look at the experimental protocol. Again, you're not including a lot of information about results or talking about anything that would be in the discussion or talking about something that would be in the abstract of a paper, just the methods, just what you did, just what occurred. Okay, and passive voice and past tense, and only for the experimental system. So now, again, the control group. What's a control group? Our positive control. Why? Because we know what the known effects are. What's our control group? Yeast and glucose. Make it pretty. Talk about it. Give me an explanation. What's the experimental group and why? Yeast and galactose, because we're testing it. It's a stereoisomer of yeast and glucose. We do not know whether or not it's going to um, produce the same or lower levels of carbon dioxide. This depends on whether or not the null or alternative hypothesis is validated, okay, and whether or not structure is found to determine function. That's why it's the experimental group. What is your prediction for the results of the experiment if the null hypothesis is valid? So, prediction, again, we're talking about means. Null hypothesis, structure does not determine function. Structure does not determine function. We expect that positive control and that, or that control group and that experimental group to have the same, basically the same, or no statistical difference between the mean production of carbon dioxide. And you need to identify what those groups are and give me an explanation for it. Okay. The alternative hypothesis that structure does determine function, we're talking about yeast and galactose, right? So if the alternative hypothesis is valid, then we are going to say that the mean production of carbon dioxide as an effect of the experimental group is statistically less than that of the what? Control group. Why? Because it's producing the same effect that would be seen in yeast and water, and that molecule cannot be used in what? Alcoholic fermentation. So, what's your deductive logic statement for the results of the experiment if the null hypothesis is valid? Oh, So again, it's just that if-then statement. If structure does not determine function, then what did you write about the prediction above? And again here, if structure does determine function, then what did you write about the alternative hypothesis for the prediction above? So again, you're going back into the methods. You're writing a passive voice, past tense, not using first person pronouns, not using words such as we did or something is occurring, past tense. And you are including the results, nothing about the discussion, or you're including the methods, nothing about the discussion, nothing about the experimental results, just what was done, strictly nothing else here. Okay? Results. So again, here it's saying to copy and paste the link for your copy of the exercise fermentation data sheet with your exercise 4 data into the text box below. Okay, so, and you've got you need to put your name, this, and your section number, and be sure to have provided your instructor access to edit your file so they can provide feedback if needed. You will notice at the bottom of the window there are two separate sheets with a singular Google Sage file. Just copy and paste it in here. Okay, and let me view the data tables. Okay, put the data tables in. All right, and you can include the name of the data table figure so that I know what you're referencing here. All right, and then here, you're going to take a screenshot of your title graph and insert it into your Google Sheets in the text box below. Okay, in the graph text box below. Okay, screenshots of the example graph provided by your lab instructor will not be accepted. So what they're asking you to do is basically go in, go here to this figure and hit your screenshot icon or your snippet tool and just grab it in, put it in there. Okay. Um, you should be able to upload this reliably in here at a point where I can see it. So if I'm not mistaken, you guys had feedback from your first digital lab notebook. Make sure you go in and you look and you know whether or not I could see it. If I could not see it, post the link, post the figure title below. Okay, make sure you have your figure title below the graph, okay, so I can see it. Write a summary of the data trends. You're writing results. Again, on As You Learn, I uploaded a document explaining how to write results. You are talking about what the results are, not what they show, what they are. What are the p-values for your experimental and controls? What are the p-values for your what? Positive and negative controls. And you can say whether or not the means are statistically different based on those values. Okay. And there's a resource there to help you as well if you need more help. Discussion. Now, what happened to the phenomenon of interest in the negative and positive controls? Talk about the amount of carbon dioxide produced. Reference the p-values. Okay. Is there a difference between what you observed in the phenomenon of interest between the negative and positive controls? Yes. If so, was it or was not this anticipated? It was anticipated. Okay. Complete the phrase below. Right. If the mean amount of carbon dioxide produced by yeast and glucose is statistically what? Greater than the mean or the milliliter produced by yeast and water, therefore the results are consistent. The experimental system was working. Therefore, we can interpret the results and we can assess the validity of the hypotheses. And you're just going to go through and answer these questions. What was the p-value for the comparison between the negative and positive controls? Identify the groups. Tell me what it was. Does this p-value indicate statistical significance between these two groups? Yes. How can you tell? It's less than 0.05. Okay? And you can go in to this sheet and again, look here. You can see where we talked about p-values. If it's less than 0.05, the two samples are different. They're less likely to occur and thus have only a 5% chance, indicating that the results are significant. Say that. So when comparing the error bars for the negative and positive controls, is the statistical significance between these two groups indicated, and how can you tell? Well, with the p-values being significant, the error bars would not overlap, okay? And that would indicate significance between those groups, okay? Would that align with the p-value? Yes. That would align with the p-value because it would show difference. Again, if the p-value is less than 0.05, it shows that they are statistically different, right? They have a very low probability to occur with overlap. Right, you can talk about that. And then testing your hypothesis. So, what happened with the phenomena of interest in the control and experimental groups? Okay, well, in the control group, if you look at the results, that was greater. Okay, it was statistically greater in the control group as compared to this experimental group. Did that align with what we anticipated to happen if the alternative hypothesis was validated? Yes. It aligned with what would occur whenever we were looking and seeing if structure determined function. Okay, because there was a difference between these groups. And remember, that is the one that is crafted based on what we thought would be answer the experimental question. And again, yeast and galactose is statistically greater than yeast and glucose. The results are consistent, or statistically, yeast and galactose is statistically less than yeast and glucose. The results are inconsistent for the prediction with the null hypothesis, and then we would reject the validity of the null and conclude that the null was invalid. Okay. Okay. And the opposite. Yeast and galactose is statistically less than the mean of the yeast and glucose. Those results were consistent with the alternative. Therefore, we would fail to reject the validity of the alternative and conclude that the validity is valid. Okay? And again, you'd answer it. What are the p-value for the comparison of the control and experimental groups? Give me those values. Does that indicate statistical significance between these groups? Yes. How can you tell? The p-value is less than 0.05, which means that they have a low probability of occurring, which means that there is significance in the difference between these values, and that if you looked at them and there were error bars, okay, you would see that the error bars would not overlap, okay? Would that align with the p-value? Yes. Again, because the p-value is showing that there is a less than 0.05, right? And that there is a low probability of these two events occurring with significance over the mean. Okay. We were able to answer the experimental question. We validated the alternative hypothesis, and it was effective for testing our experimental group, and you can explain why. Again, talk about p-values, talk about why it was evaluated, talk about the purpose of the experimental and or the positive and negative controls, what their functions were, and why that was important, and do the same here. Okay? Do not forget to put your name down here. If you do not, that will be a 0. You will lose 5 points. So, yeah. Any questions about the DLN? Any questions at all? One thing I did want to mention to you guys is that I'm going to provide some instructor provided extra credits, probably going to be about 20 points. You'll get an email sometime. Please complete this before November the 8th, okay? That way you can have your points in. That way you can ease some of your anxieties. And, you know, if you feel comfortable with where you're at with this material, you can go ahead and go. The meeting for today is ending, so I'll let you guys head out. I hope you have a good day.
I have a really quick question. For how are we supposed to describe the methods we used? Is it just do you just want us to describe what Parks did in the video?
So if you actually look, and I'm going to screen share again for those I'm recording this for, because again, there's been a couple of issues with the internet connection here recently, and I had a couple of students who were confused. But if you go in here, you know, you're just taking basically this information that's right here. You're putting it in sentence form. Okay, instead of you will, you could say a micropipette dispenses 6 milliliters of yeast solution into a fermentation tube. You changed the micro pipette tip was changed to add in 6 milliliters of the appropriate blah 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 to this, and then you could put all the ingredients were added into each of the tubes. The tube was flipped backwards to fill the stem completely with liquid. This was done several times. This was done for all 3 tubes. Each of the tubes were placed in the 37-degree water bath for 15 minutes. At the end of 15 minutes, graduations on the stem of the tube were used to record the milliliter of CO2 produced by each sample. These values were recorded. So, yeah. Cumulative results for the class were used to evaluate the results of the experiment, the overall results of the experiment. That's basically, how you would want to kind of format that method section. And again, just to kind of show you if you're worried, you can go on As You Learn. I upload this every semester when I teach 1801 because I think it's really important for students to have